Morning, everybody. Thank you so much for joining for our ZOOMinar series. Today, our guest speaker is Professor Andrew Lieberman. Thank you so much for coming, Andrew. Professor Lieberman is a Gerald Aberson Colleague Professor of Pathology and Director of Neuropathology in the Department of Pathology, Michigan Medicine, University of Michigan. And he's also a co-director of a great initiative which has been running for some 6-7 years, and it is to study the protein folding diseases. It's a really great initiative which united a lot of people from different areas and backgrounds. Dr. Lieberman received his bachelor's degree from Duke University and his MD PhD degree from the University of Maryland Medical School with mentor Dr. Moon Shin. He completed residency and following training in neuropathology at the University of Pennsylvania, and then he trained as a postdoctoral fellow with Dr. Kenneth Fishback at Neurogenetics branch at NIH. He's a recipient of Beeson Award in Aging Research and Neuroscience of Brain Disorders Award from McKnight Foundation. And he's also elected a member of the American Society of Clinical Investigations. Dr. Lieberman's research has focused on the mechanisms of neurodegeneration in inherited neurological disorders, and his laboratory uses cell and mouse models to explore the pathogenesis of Kennedy disease, which is polyglutamine expansion disorder, and Neumann-Pick C disease, which is a lysosomal storage disease, which results from impaired lipid trafficking. I don't want to keep your attention more, keep away your attention from his talk. I just want to remind everybody that the presentation will be about 35 to 40 minutes, which will be followed by a Q&A session. And once the talk finishes, this time you will be allowed to personally and directly ask your questions. Just click on, the hand icon, so you are flagged, so we will be able to let you in to ask the questions. After 11 a.m., we will read the questions from a Q&A box. Just as a reminder, please keep your questions short and concise and easy to understand. Also, another reminder that starting from September, the ZOOMinars will be at 10 a.m. Eastern Time every Saturday. We are moving them from Tuesdays to Saturday. Thank you so much for your participation and looking for your questions and feedback. And thank you, Andy, for coming. Just looking forward to your talk. Thank you.
Thanks, Magda, and thanks so much to the organizers for inviting me to participate in the ZOOMinar. I think it's a really cool idea to bring everyone together during this time when it's so easy to feel dispersed and isolated. So thank you all for organizing this. So this morning I want to tell you about some recent work in my lab studying one of the polyglutamine expansion neurodegenerative diseases. So this is a group of 9 inherited neurological disorders, all of which are caused by microsatellite repeat expansion. So the repeat is a CHE repeat. These microsatellites occur in the coding region of disease-causing genes that are otherwise dissimilar. And these genes are widely expressed throughout the brain and indeed often throughout the body, yet each of these disorders is characterized by distinct neuronal vulnerability and neuronal death in different populations. These are all chronic progressive degenerative disorders that tend to come on in midlife and are slowly progressive. They're all characterized by genetic anticipation, wherein through transmission, these tracts can increase in length, and the increased tract length can result in earlier symptom onset. And for all of these disorders, it seems that the polyglutamine expansion results in a toxic gain of function that is really a dominant driver of disease pathogenesis. But bear in mind that the mutation also can affect normal function of the disease-causing gene, resulting in a partial loss of normal function. So it's likely that the phenotype is a mixture of both of these consequences. So the disease that we study is called spinal and bulbar muscular atrophy, or SBMA, also known as Kennedy's disease. This is actually the first polyglutamine expansion disorder that was identified genetically. It's a disease of lower motor neurons and skeletal muscle, manifests in men, causing weakness and atrophy in the muscles of the upper arms and legs, and swallowing muscles in the neck, the bulbar muscles. The disease-causing mutation is in the androgen receptor, which is the male steroid hormone receptor. And because toxicity, protein misfolding of the androgen receptor with the long polyglutamine tract is dependent on hormone binding, and nuclear translocation is similarly dependent on hormone binding. And we now know that these are critical steps in disease pathogenesis. The disease actually only occurs in men who have the mutant allele. And it's a relatively rare disease affecting about 1 in 50,000 men, though precise numbers are often difficult to come by. So a number of years ago, we set out to generate a knock-in mouse model of the disease. And I'm going to show you lots of data from these mice. I just want to provide a little foundation for you to understand the phenotype that they develop, which is really robust and nicely mimics several of the manifestation of SBMA in patients. So the mutant male mice develop diminished body weight as a function of age. They have frank muscle atrophy, and that's shown here by measuring the weight of a disease-relevant hind limb muscle, the tibialis anterior. The male mice, like SBMA patients, have diminished fertility, and they have frank testicular atrophy. Again, that's age-dependent. And they have neuromuscular dysfunction that we can measure either by grip strength or by running the mice on a treadmill. And again, it's age-dependent. And all of these phenotypes are hormone-dependent. So if we surgically castrate the mutant mouse, we're able to rescue these many deficits. And in studying this mouse model, we've come to learn that this isn't just a disease of lower motor neurons, but rather skeletal muscle seems to be a very important contributor to SBMA pathogenesis. And we began to appreciate this by studying the pathology that develops in these knock-in mice, and found that the skeletal muscle shows really profound pathologic changes months before any detectable disease was evident in the spinal cord. So the kinetics of the disease suggested an important role for skeletal muscle. And this was really supported by work that we did in collaboration with Ionis Pharmaceuticals, where we treated mice with androgen receptor targeted antisense oligonucleotides. These were delivered subcutaneously, and through this route, they don't cross the blood-brain barrier. So we were only knocking down peripheral expression of the polyglutamine androgen receptor and not expression in the lower motor neurons, and found that this route of administration significantly reduced all of the disease manifestations in the mice that we had ever followed. And this observation was really corroborated by work in a back transgenic mouse. model of SBMA, where conditional deletion of the polyglutamine androgen receptor only in skeletal muscle similarly reduced disease phenotypes. And so together, these data really suggest an important role for muscle in the disease phenotype in several different mouse models. Similarly, there's been strong evidence in patients with SBMA that muscle is an important contributor to pathogenesis. And in fact, in a series of recent studies looking at biomarkers, it's been found that these patients do not show increased neurofilament light chain, which is a nice biomarker that has been used in a variety of neurodegenerative disease settings to study axonal damage, but rather shows increased levels of creatine kinase, a marker of muscle damage. So based on these and other data that's in the literature, we've come to think of Kennedy's disease, or SBMA, not as a disease of motor neurons or just a disease of skeletal muscle, but rather a degenerative disorder that affects the neuromuscular system, where both of these compartments, both the lower motor neurons and skeletal muscle, express the polyglutamine androgen receptor. Early in the disease, the muscle symptomatology seems to be quite profound and drives some of the initial manifestations. But as the disease progresses, and certainly by end stage, there's frank lower motor neuron degeneration. And at that point, it's quite likely that the lower motor neuron component predominates. So what I want to talk to you about today are two areas that we've been really focusing on of late. The first is looking at the mechanisms that drive skeletal muscle toxicity in SBMA. And we're compelled to do this for several reasons. First, the mechanisms are poorly understood. So there's, I think, substantial biological interest there, but also skeletal muscle represents an attractive therapeutic target that's easier to access than lower motor neurons. And I think could quite likely impact the clinical phenotype. And then towards the end, I'm going to switch and talk about the strategy that we're using to target molecular chaperones as a potential therapeutic. So we began trying to understand the mechanisms by which the polyglutamine androgen receptor causes skeletal muscle pathology simply by looking at gene expression changes in either our knock-in mice or wild-type controls that were evident on RNA-seq analysis. And quite unexpectedly, what we found from these initial studies was that there was a marked decrease in the expression of a whole slew of genes that encode components of the ubiquitin proteasome pathway. So this included about 30% of all of the genes that encode proteasome components, as well as 20% of the E2 ubiquitin ligases. So in this diagram here, you can see the many proteasome genes that were downregulated, detected to be downregulated in the SBMA mice. This included components of the 20S core as well as 19S cap. And this pathway really attracted our attention because of the fact that data in the literature clearly indicated that the polyglutamine androgen receptor, particularly in the nucleus, is degraded primarily through the proteasome. So we knew that that the polyQAR, when it misfolds, is ubiquitinated through these some of the very ligases that are downregulated in the disease and then sent to the proteasome for degradation. And it seemed quite reasonable that downregulation of this pathway could exacerbate toxicity by increasing the steady state levels of the mutant protein. So we set out to determine the functional consequences of proteasome downregulation. And this is just simply qPCR data looking at mice at both early and later time points. And I'm showing 3 different genes that we identified to be downregulated. One is a scaffold protein of the 20S proteasome. The middle one is component of the 19S lid, and the third is one of the E2 ubiquitin ligases. You can see that they're all down in this pelvic floor muscle that expresses high levels of the androgen receptor at an early time point. We also looked at two different hind limb muscles, the quadriceps and the tibialis anterior, and in both of these cases, these muscles were only slightly affected or not significantly affected at early time points, but as the disease progressed, these proteasome components were significantly downregulated at the RNA level. Similarly, they were found to be robustly downregulated at the protein level. So these are protein lysates from quadricep muscle from the mutant mice compared to littermate wild-type controls. And you can see that a whole slew of proteasome components are significantly diminished in the SBMA mice. Not only is the proteasome decreased at the expression level, but similarly, proteasome activity was found to be functionally impaired. And we tested this using several different assays. I'm just going to show you data from one of those, and that is using one of these activity-based probes. So these probes have a reactive group at one end that covalently binds to the catalytic domain, the catalytic subunits of the 20S proteasome when they're assembled into a core. So these bind to the trypsin or chymotrypsin-like activity subunits of the 20S proteasome. And at the other end, they have a reporter tag, and in our case, we used fluorescently labeled or BODIPI labeled activity-based probes. So you bind the probes to the proteasomes and lysates, and then because they're covalently bound, you resolve them on polyacrylamide gel. And you can see quite nicely the catalytic subunits in wild-type mice and significantly diminished activity for both trypsin and chymotrypsin-like activities in the SBMA mice. So just to summarize what I've shown you so far, what we found is that there is, in fact, reduced expression of proteasome subunits at both the RNA and protein level in SBMA mice. There's reduced proteasome activity as measured by several assays. I just showed you data from the activity-based probes. We also, interestingly, we found that of those proteasomes that are intact in the muscle, they are actually post-translationally modified to be inactivated. So this subunit, ADRM1, is the subunit of the lid that binds ubiquitinated substrates as they're being recruited in to the proteasome for degradation. We found that these subunits are significantly ubiquitinated themselves in the SBMA mice. This ubiquitination impairs the binding of substrates to the proteasome, so post-translationally inactivates them. Then using both cell and fly models, we were able to show that these modifications enhance polyglutamine AR toxicity. The curious thing about this, or the surprising thing, is that we also know that proteasomes are really critically important for muscle atrophy. Yet we were seeing this impaired proteasome function in the context of SBMA muscle atrophy. And just to highlight this, these are data looking at muscle atrophy from a variety of causes, including denervation, spinal cord injury, tumor bearing, uremia, diabetes, even in ALS models. And what has been shown is that there is increased activation of the ubiquitin proteasome pathway in many or in all of these settings. Yet our analysis had unexpectedly shown diminished activity of this pathway. And this raised the question for us, well, if that's the case, then how is muscle atrophy occurring in SBMA? And so we set out to very systematically evaluate the many different steps that might regulate the steady state homeostasis in a muscle. So we knew already when we started this that there was diminished activity of the ubiquitin proteasome pathway. So that was down. We then went all the way back sort of to the stem cell level and looked at factors that affect muscle stem cells to see if those were abnormal. And what we found was that actually the signals for making muscle, activating muscle stem cells, were actually increased in the SBMA mice. And similarly, the inhibitors of muscle growth, such as the myostatin signaling pathways, are actually decreased, thus setting up a situation where one would favor muscle hypertrophy rather than muscle atrophy. We also tested the functional capacity of the stem cell niche by injuring the muscle and looking at its capacity to regenerate, and found that the SBMA muscle regenerated just as well as wild-type muscle. So all of this then really begged the question for us as to where does the muscle go in SBMA? Why are these patients, why are the mice experiencing such profound muscle atrophy? And so to answer this question, we went back to our RNA-seq data set, and what we found was that of the top 5 Go terms of genes that are down-regulated in the SBMA mice, all of them encode components of sarcomeres. And while this might have been expected for sort of a proteomic analysis of atrophic muscle, it was a bit surprising to find this to be the case in a RNA-seq analysis of atrophic muscle, and prompted us to ask whether or not there was a critical transcription factor that might be regulating these genes and whose function would be impaired. And so that's what we did. We used a program called TopGene to look at the transcription factor binding sites, or the predicted transcription factor binding sites, in the set of genes that was altered in SBMA muscle. And using this in 2 different RNA-seq data sets, the one for the quadriceps and the other for that pelvic floor muscle that expresses high levels of AR, what we found was that the top 5 hits for transcription factor binding sites all were for this transcription factor known as MEF2. So MEF2 is a very well-studied transcription factor that plays a critical role in sarcomere homeostasis and in driving muscle hypertrophy. And so it's really interesting to us that this transcription factor was sort of nominated by our TopGene analysis as being critically dysregulated in the disease. So as I mentioned, MEF2 has been studied in a whole slew of different settings. So there were RNA-seq and in fact ChIP-seq data sets of genes that in muscle that are responsive to the knockdown of MEF2. So we simply ask, what is the overlap between the genes that are known to be MEF2 targets versus the gene expression changes that we've found in SBMA muscle, either the pelvic floor muscle or in the quadriceps. And what we see is that between 20 and 40% of the genes that are MEF2 targets are similarly altered in the SBMA muscle, resulting in very high p-values. Similarly, there are other data sets in the literature in which MRF4 is knocked down. So this is an endogenous inhibitor of MEF2. So it results in so knocking down MRF4. This inhibitor results in MEF2 activation. So again, we looked at the set of MRF4 knockdown responsive genes in skeletal muscle, and once again found between 10 and 30% of those genes are overlapping with the gene expression changes in the SBMA muscle. And just another way to look at this is shown here in these heat maps. Compared to wild type, you could see this set of MEF2 regulated genes that are altered in the SBMA muscle. This is rescued quite nicely by treating the SBMA muscle with antisense oligonucleotides targeted against the androgen receptor. So you see we get a nice rescue of that, really demonstrating that this is driven by polyglutamine AR proteotoxicity. And similarly, you see a very similar pattern between this ASO and knockdown of MRF4, this knockdown MR4, which triggers MEF2 activation. So we think that there's impaired MEF2 function in SBMA muscle. And to look at this more directly, we electroporated into the muscle of living SBMA mice a plasmid encoding MEF2 binding sites driving luciferase. So we did a luciferase activity assay in vivo, collected the muscle 48 hours after we electroporated the animals, and then looked at luciferase activity, and you can see it was significantly lower in the SBMA mice than in wild-type controls. Now, this was interesting to us since we knew from looking at our RNA-seq data set and by independent Western blot confirmation that MEF2 protein levels were unaltered in the SBMA mice. And this prompted us to ask whether or not their intranuclear localization may be different. So this is the intranuclear localization of MEF2, diffusely present within the muscle nucleus in a control mouse. In the SBMA mice, we see this really robust sequestration to P62 positive intranuclear inclusions that are robustly present in the SBMA muscle. Not only were MEF2 targets down in SBMA mice, but we also found that they were down in patient biopsies from SBMA patients. So this is looking at 4 of about half dozen or so different MEF2 targets that we studied in SBMA patients, and you can see that all of them are significantly down in this cohort of individuals. And finally, we asked whether restoring MEF2 activity would have beneficial effects in the SBMA mice. And to do this, what we did was we electroporated into the mouse muscle plasmids encoding either a constitutively active MEF2 or MEF2 with a DNA binding domain. So a DNA binding domain mutant that's functionally inactive. And you can see that when we express the constitutively active MEF2, we were able to significantly rescue muscle fiber size. The effects are quantified on the right here. And we were able to rescue MEF2 target gene expression. So these are the same genes that were downregulated in the SBMA muscle, patient muscle. They're down also in the SBMA mice, and their expression is largely rescued by expressing the constitutively active MEF2. So just to summarize this part of the talk, what we found was that SBMA muscle shows a lack of ubiquitin proteasome-based atrophy, that type of atrophy that's seen in ALS models and many other models of skeletal muscle atrophy. We looked carefully for signals that affect muscle growth and found that they're altered to favor muscle hypertrophy. We've evaluated the muscle stem cell niche and found that it's able to successfully regenerate after injury. But quite unexpectedly, what we found as what we think is a really critical driver of muscle atrophy in this disease is that MEF2 function is impaired, and it's functionally sequestered into these intranuclear aggregates, leading to diminished expression of MEF2 target genes. We see a similar loss of MEF2 targets in SBMA mice and in patients, and we're able to restore MEF2 activity using this constitutively active mutant that rescues atrophy and restores gene expression. So we're continuing to be quite interested in the potential consequences of restoring MEF2 activity in the long term, and those are ongoing experiments that we're just getting ready to start in the lab. Okay, at this point, I want to just switch focus and talk about one other area that's of really active interest in the lab, and that is some studies looking at the role of molecular chaperones as potential therapeutic targets. And we became interested in this based on a large amount of data in the literature not only in SBMA, but for all of the polyglutamine disorders, where many really excellent labs using valid model systems have found a whole slew of pathways that are dysregulated in disease. So there's evidence for transcriptional dysregulation, as I just showed you, for MEF2 in muscle and SBMA, but other transcription factors have been implicated in disease pathology and other polyglutamine disorders. There's also evidence of cytoskeletal abnormalities, mitochondrial dysfunction, proteostasis impairments. And the fact that there are so many different downstream targets that are dysregulated in the disease suggests that from a therapeutic standpoint, targeting any single one of these may be insufficient and to rescue the full spectrum of disease phenotypes. And so again, from a therapeutic point of view, this has really focused our attention on sort of the polyglutamine protein itself as the proximal mediator of toxicity. And it's prompted us to think about strategies to get rid of the polyglutamine androgen receptor. And the strategy that we have focused on really takes advantage of the endogenous cellular machinery that normally regulates AR proteostasis, and that is the HSP70, HSP90 based chaperone machinery. So unlike the classical notion of chaperones, individual chaperones working to promote the folding of newly synthesized proteins, this multi-protein chaperone machinery instead binds to proteins in their native or near native conformation. So it's interacting with mature proteins, and for the steroid receptors, including AR, it's been shown to regulate a whole slew of important activities that govern its normal function. So for the steroid receptors, interaction with HSP70 and 90 is required to open the ligand binding cleft to enable steroid to enter into the ligand binding cleft of the steroid receptor. There's evidence that the chaperone machinery tethers the steroid-bound receptor to the microtubule apparatus and promotes cytoplasmic to nuclear translocation. And then when these receptors are either damaged or toxically misfold, the chaperone machinery plays an important role in degrading the mutant protein. And it's this action of the chaperone machinery in protein quality control that's really attracted our interest. And so we've developed a model in which, as the polyglutamine androgen receptor misfolds, its interaction with HSP90 is lost, and that leaves HSP70 to recruit in chaperone-dependent E3 ligases such as CHIP, which then ubiquitinate the polyglutamine androgen receptor and send it for degradation by the proteasome. So that's the model that we're studying. I just want to show you evidence, experimental evidence from the lab that really validates the fact that the androgen receptor is a classic HSP90 chaperone machinery client. So here what we've done is we've taken lysates expressing either the wild type or the polyglutamine androgen receptor and immunoprecipitated using immune or non-immune serum. So when we pull down the androgen receptor from these lysates, we co-precipitate HSP70 and HSP90. These are actually functional AR chaperone machinery complexes, as evidenced by the fact that we're then able to use these complexes that are isolated and bind radiolabeled ligand. We can then strip off the chaperones. We get a nice strip, and this abrogates ligand binding activity, and then we can rebuild the chaperone machinery on the isolated androgen receptor, either using rabbit reticulocyte lysate or purified components of the chaperone machinery. Either case, you can see that we restore 70 and 90 binding, and this reconstituted complex then once again binds ligand. So the AR is a classic client of the 90-base chaperone machinery. And based on the model of proteostasis that I showed you, it really suggested to us that 70 might be a good therapeutic target for degrading the misfolded androgen receptor. And this idea is supported by a lot of literature, genetic literature, in which either HSP70 or its co-chaperone HSP40 are overexpressed in a whole slew of neurodegenerative disease models that have been constructed from yeast to mouse, many of which overexpress HSP90 client proteins, not only SBMA, but also tau and Alzheimer's disease, Huntington, and synuclein, all of which are known to be clients of the 90 chaperone machinery. So to approach whether or not we could target HSP70 therapeutically, we took advantage of the fact that that 70 has ATPase activity. So the protein has two functional domains, a nucleotide binding domain and a substrate binding domain. When 70 is bound to ATP, this lid and the substrate binding domain is open and it binds to substrates with so-called low affinity, whereas in the ADP bound form, the lid is closed and substrate binding is at high affinity. So what we sought to do was favor the ADP bound state of HSP70 to promote binding between the misfolded androgen receptor and HSP70 in order to promote its ubiquitination and clearance. And we did this work in collaboration with Jason Guswicki, who's a chemical biologist who was at Michigan at the time, now at UCSF, Yoichi Izawa, colleague in pharmacology, and Bill Pratt, who was also in pharmacology. And what we found was that using either chemical or genetic approaches to favor the ADP-bound state of HSP70, we're able to, in fact, promote ubiquitination and clearance of the polyglutamine androgen receptor. So the genetic approach that we took was to overexpress the co-chaperone HIP, which is known to favor the ADP-bound state of HSP70. And then as we were conducting those genetic experiments, Jason's lab identified a small molecule called YM1 that acts similarly to HIP to favor the ADP-bound state of 70. And by performing these manipulations, either in cell culture or in flies, we found that we were able to promote ubiquitination of chaperone machinery clients and clearance of the polyglutamine AR to rescue disease phenotype. More recently, we've collaborated with Jason and Javier Sabatia and found that HSP70 not only interacts with the ligand binding domain of the steroid receptor and as a component of this chaperone machinery, but separately interacts at a site at the amino terminus of the androgen receptor, which is just upstream of the polyglutamine track. And interestingly, this interaction site includes a sequence that's known to interact with a sequence at the C-terminus of the androgen receptor, and that this NC interaction occurs in a hormone-dependent manner. So we think that in the absence of hormone, 70 binds there, prevents that NC interaction. In the presence of hormone, 70 comes off, enabling that NC interaction. So we think it has an important regulatory role in its normal function, but binding there also can prevent self-oligomerization of the misfolded androgen receptor. And so we took a derivatized compound of YM1 that Jason's lab made to try to stabilize the interaction of HSP70 with the polyglutamine androgen receptor in mice. So we're stabilizing either at the ligand binding domain or at the amino terminus. We don't know which site is really critical for this, but in either case, what we see is a nice rescue of polyglutamine AR levels. So these are lysates from mice that were treated for 2 weeks with either vehicle or a compound called JG294, which is another derivative of the YM1 family. See, after drug treatment for 2 weeks, there are significantly lower levels of monomeric polyglutamine androgen receptor, as well as diminished amounts of these high molecular weight species, these smears that appear at the top of the polyacrylamide gel, you can see that the oligomeric species are really nicely visualized on agarose gel. There's clear diminution following treatment with JG294. Similarly, we see decreased staining for androgen receptor in sections of skeletal muscle after mice are treated with JG294. And all of this clearances occurs without any change in polyglutamine AR mRNA levels, really consistent with the model that we're promoting clearance of the mutant protein. And finally, I just want to make one last point about the strategy of targeting 70 versus HSP90. So there's been a lot of work on HSP90 inhibitors. A good number of them have now advanced to clinical trial in humans, largely for cancer treatment. One of the challenges with targeting HSP90, or there are a couple of challenges. One is that they robustly induce a stress response. And the second is that interaction of client proteins with HSP90 largely stabilizes those client proteins. So if you inhibit HSP90, you not only degrade your target protein of interest, but you can also degrade other clients that are in their native conformation and are needed for cellular function. So here we've treated mice with either JG294 or DMAG17. This is a nice, well-tolerated HSP90 inhibitor that we can give to animals. You can see that the 90 inhibitor robustly induces a stress response, as evidenced by this induction of HSP70, and it also diminishes the expression of other clients. In this case, we're looking at ERK, that are in their native conformation that are needed for normal cellular function. And neither of these occur when we activate 70-dependent ubiquitination and degradation of the polyglutamine androgen receptor. So we think that this strategy has potentially great advantage over the more commonly used strategy of targeting HSP90. So just to finish up this part of the talk, what I've shown you is evidence that the HSP90-70-based chaperone machinery regulates AR proteostasis, including both ligand binding and degradation of the misfolded polyglutamine androgen receptor. We're taking a strategy of trying to favor the ADP bound conformation of 70 to increase its activity in promoting ubiquitination and degradation of this mutant protein. We've identified some small molecules that are active in cellular models and in mice. And we are really actively pursuing this now because we think that this could be a nice way to target the protein quality control pathway while avoiding some of the toxicities that are inherent in targeting HSP90. So just to finish up, I want to just acknowledge the people in my lab who did the work that I showed. Those people are highlighted in bold here. So Sam Nath is a MSTP student whose thesis work really defined the impairment of ubiquitin proteasome pathway and diminished function of MEF2. Goritza and Varuna are former postdocs in the lab who have worked quite extensively on the Chaperone machinery project, and I showed their data. And Zhigang is a research associate in the lab who's done a lot of the work on our SBMA mouse model, including treatment with antisense oligonucleotides. This work was done in collaboration with Ionis Pharmaceuticals, particularly Jean Hung. Our collaborators in Italy, Maria Panuto, provided analysis of the human SBMA tissue. And then I have a really great set of collaborators, both here and at UCSF and internationally, who have helped us out on the HSP70 project. This includes Jason Kiswicki at UCSF, Yoichi Ozawa here at Michigan, Javier Sabatia, and I have to acknowledge the really strong intellectual contributions of our this recently deceased colleague, Bill Pratt, whose work on the Chaperone machinery has been incredibly impactful for us. So that's it. Thanks very much for listening, and I'm happy to answer any questions.
Thank you, Andy. It was such a great talk. I'm just looking to see, give a little bit of a time for the questions to come. Maybe I can ask my question.
Sure.
So, Andy, in this disease, mostly skeletal muscles are affected, right? The smooth muscles are not affected, or it eventually affects everything. I might have missed that.
So the disease in patients and in the mice, I think it starts out pretty markedly in the muscle, but it progresses clearly to involve lower motor neurons. And by end stage in patients, there's frank lower motor neuron degeneration. But it does not progress. It's not like Huntington disease, where at end stage in some Huntington patients, it's not just the striatum, but it's diffuse neurodegeneration throughout the brain. That's not the case in SBMA.
So all muscles are affected too. The smooth muscle, the skeletal muscle, the cardiac, all of them.
So it's mostly skeletal muscle, or it is skeletal muscle predominantly, and the proximal muscles in particular, meaning the upper arm and leg, and the swallowing muscles of the neck. There is some evidence that SBMA patients develop some cardiac arrhythmia. This is work from Ginny Suaro, but we have not detected very high levels or any cardiac pathology in the SBMA mice, and I'm unaware of any effects in smooth muscle.
So Andy, have you thought of looking into these differences? Why do you think some muscles are affected more than the others?
Yeah, that's a really great question. Right, why proximal versus distal? Why bulbar muscles? I think it's another variation of the selective neuronal vulnerability question that is so prominent in the CNS. And has been so difficult to sort out. And I think it's likely a combination of factors. Probably expression level is important. And there may be other critical protein factors that are expressed in these target cells that are not present in cells that are spared from disease, so there could either be a risk protein or a protective protein that contributes to selective vulnerability. But it's an outstanding question that we don't know the answer to.
Thank you. I think we have incoming questions from Wang.
Mm-hmm.
I
so
see.
Magda, can I promote Wang to?
Yeah. But his microphone looks muted.
I'm going to do that. 1 second. I'm going to enable him to. Where's Wang?
And I just want to remind everybody, you can raise your hands. I start seeing raised hands. And I see the type questions, so we will go first through the raised hands, and we'll leave the questions which are on queue.
So Wang, you can ask the question directly to the speaker. Wang, are you there?
Can you hear me?
Yeah.
Yeah.
Hi, Andy. Very nice talk. Just curious, how I'm interested in cell biology.
Mm-hmm.
So how does this, polyQ-AR affect proteasomes versus autophagosomes? What's the balance, how it works there, how the HSPs coordinate with those factors, and the aggregation of the what's the transcription factor you have is selective, or is it just for this MEF2, right? So is that selective for this transcription factor?
Okay. Wow. So many great questions. Lots to poke at here. So what we've found and others have found is that although there's clear dysregulation of the ubiquitin proteasome pathway, particularly in muscle where we've studied it the most, we see upregulation of macroautophagy. And perhaps that's a compensatory upregulation of macroautophagy. And in that case, in that setting, if we impair autophagy, it actually is a bit beneficial in that we're able to slightly decrease muscle atrophy. So it seems that the autophagic pathway is likely to be upregulated as a compensatory way to handle a whole slew of misfolded proteins. And it's upregulated to such an extent that it may have some detrimental effects, at least in this disease setting. In terms of MEF2, we think it's a really interesting target. I doubt that it's the only transcription factor that is sequestered into these aggregates. There's evidence in other polyglutamine diseases and in other models of SBMA, particularly cellular models, that other transcription factors can be sequestered as well. I think the functional importance of MEF2, particularly as we've seen in skeletal muscle, is quite profound. So I think it's got clear biological relevance as we're able to rescue muscle atrophy by overexpressing a constitutively active form of MEF2. But I wouldn't expect it to be the only transcription factor that's there. I didn't show the data, but we also know that MEF2 is sequestered into aggregates in the R62 model of Huntington disease. So those mice express just a fragment of the polyglutamine Huntington protein. They develop severe muscle atrophy. And so we were really interested to see whether or not there was specificity in terms of, is it the androgen receptor that is required for this aggregation and loss of function, or is it sort of expanded polyglutamine tract? So we examined those R62 mice fairly carefully and found similar overlap by RNA-seq with a whole slew of MEF2 targets, and similar sequestration of MEF2 into the polyglutamine aggregates in R62 muscle.
So we have Bin Zhu. You can ask the speaker question directly. Bin?
Yes.
Yeah, go ahead.
Very nice talk, Dr. Lieberman. Very interesting. So I have two questions. First is, since your the polyglutamine protein, whether it has some similarity or difference with the other well-studied protein, Huntington, and whether your finding of HSP70 or MEF2 were applicable to Huntington gene as well. And the second question regarding your therapeutic part of your talk, whether you have considered other approaches in addition to your HSP70 stabilization, looking at like aggregation inhibitors or inflammation inhibitors, things like that, because there is a variety of approaches to try to reduce the neurotoxicity of those aggregation proteins.
Right. So great questions. Regarding Huntington disease, I think that as I mentioned to Wang, we know that MEF2 is aggregated into the internuclear inclusions in muscle of the R62 mouse, and seems to functionally impair MEF2 in that Huntington model, similarly to what's happening in the SBMA mice. And so we think that this is being driven by protein misfolding mediated by the expanded polyglutamine tract that's similar between SBMA and Huntington's disease. And, you know, that's pretty much where things stand right now. I would note that there are a couple of other papers in the literature that have shown MEF2 dysfunction in polyglutamine models. There was a recent one looking at impaired MEF2 function in the hippocampus of Huntington disease mice. And there's a prior paper, I think, in a SCA model in cell culture looking at impaired MEF2 function. So I think it is a transcription factor that has a intrinsically disordered domain, and because of that is nicely recruited into these, or is potently recruited into these polyglutamine aggregates or soluble species of the misfolded polyglutamine proteins, leading to functional impairment. Regarding other strategies in addition to HSP90 activation, our other sort of foray into treating the SBMA mice has been in collaboration with Ionis, testing their antisense oligonucleotides. We have not tested any aggregation inhibitors or anti-inflammatory strategies, but you know, I think that those would be reasonable things to try. Thanks for the question.
Magda, you can continue.
I think it's very close to 11, so we can.
There's one question from anonymous attendee. You can ask that
question
in the Q&A folder.
Yeah. How effective would be the transcription factors as therapeutic agents compared to small molecules? What will be the challenges when working with the transcription factors?
Yeah, so that's a interesting question. I think that there are lots of challenges while working with transcription factors as a therapy. So one presumably needs to restore their function at an appropriate level so that, and do it in the context that its activity can be appropriately regulated. So sometimes you don't want too much of a good thing. So that's one set of challenges. The other is, of course, simply delivery. Where a small molecule, that path to delivery is more easily envisioned. One would have to perhaps take a gene therapy approach to delivering a transcription factor. I think it's possible to try it, and something that we're very much considering with MEF2 and having it attached to putting on some like a tamoxifen regulated domain, so that we could so it isn't always on. So we could turn it on, turn it off, and hopefully avoid the challenges that might occur from chronically active MEF2. But we, you know, that's sort of in planning stages and has not yet been tried.
I have one question. So like in one of your slides, you have shown that the HSP70 is kind of ATP independent pathway. It can degrade the poly-Q-AR, right? So, but like, can you hear me?
Yeah, I'm sorry. I didn't hear your can you repeat your statement?
Yeah, like you have shown that like the HSP70 works in a ATP independent manner to degrade the poly-Q-AR, right?
ATP independent.
ATP independent because it happens in the ADP bound state.
Yeah, well, it's nucleotide bound, so I don't know that I would say it's ATP independent. What we're doing, and we're not blocking the ATPase cycle. I think that's a different kind of strategy, or we're not. promoting the cycle. We're simply maybe the way I think about it is that we're slowing the cycle between an ATP bound state and an ADP bound state, and thereby favoring the allosteric conformation that binds to misfolded proteins with high affinity. So we're simply favoring that state rather than blocking it.
Okay. I think in the human prion studies, people have shown that the co-chaperone activity, like when you have HSP 104, 70, and 40, they work in a better way like a single chaperone. So in your case, do you think that if you have like different kind of chaperones mixed system, the degradation can be activated in a more profound way?
Yeah, so I would imagine it is going to depend on whether or not the client, in our case, polyglutamine androgen receptor, in the prion disease, I guess, prion protein, whether the client protein is a client of the HSP90 70 base chaperone machinery. So we know that AR is, and Huntington, there's really nice data showing Huntington is, and tau, synuclein, these are all clients of the 90 chaperone machinery, and that's what prompted us to study it, not as individual chaperones, but in the context of this machinery. I don't know if there's any literature to indicate whether or not the prion protein is a client. I would think it might not be, just, but I actually don't know that literature.
Okay.
We have one question coming from Martin Muschel.
Martin can ask question directly.
Yeah.
He has access now.
Yeah, I see him.
Martin,
listed.
go ahead.
Yeah.
Hello?
Okay, am I audible?
Audible, yeah.
Okay, great. Yeah, I wasn't quite sure if I caught that. I wasn't sure, do you know what type of aggregates are formed by the polyQ-AR in the muscles? Are they fibrillar in structure? Are they oligomers, or you're not sure? And what's their predominant location in the muscles themselves?
Yeah, so what we know right now is that the muscles exhibit intranuclear aggregates of the androgen receptor that are ubiquitinated and P62 positive. We don't yet know what they look like under EM or after biochemical isolation, and that, you know, hopefully we'll know that in a year or so, but I don't know that yet. But I think it's an important question that we're just starting to study. Yeah.
Fascinating story.
Yeah. Thanks.
Thank you.
Okay. Thanks.
So I think it's almost time before we transition to just closing the session. There's one more question from anonymous attendee who asked, how does being an IDP help the intake of transcription factors? And this is the last question. After that, we can all rejoin to just continue discussions with Dr. Lieberman.
How does being an IDP, can you help? What is IDP? Help me out.
I think IDP intrinsically disordered protein.
Got you.
Like it's probably referring
intrinsically
to
disordered protein. Yes, of course.
Yeah, uptake the transcription factors. Yeah, sorry, we have acronyms too.
So this is speculation. Well, it's a little bit of speculation. There is a tiny bit of data behind it. So many of the intrinsically disordered proteins are known to either be sequestered in aggregates or undergo phase transition. Using some prediction software, we found that MEF2 has a large domain that is intrinsically disordered and is sequestered into polyglutamine AR aggregates. When we swap out that domain, so we made that constitutively active form of MEF2 instead of having its endogenous intrinsically disordered domain has the VP16 transactivating domain. It just turns out that when we put that constitutively active mutant in prediction software, it has a much smaller region of predicted intrinsic disorder. And interestingly, that form of MEF2 is not sequestered into aggregates, but is diffuse within the cytoplasm of cells. So I think that so it's based on that sort of very initial, I would say, analysis that I made the comment that perhaps the intrinsic disorder domain is important for sequestration, but certainly warrants a lot more study than what we've done.
Thank you, Andy. So now we can continue join on the session. So who wants to stay? We can participate in the further discussion.
Okay.
Andy, it was a great talk. I really
Yeah,
enjoyed
thank
very
you so much, Andy.
A lot of learning
Yeah.
to me. So in the aggregation of AR, androgen receptor protein, are there any other amyloid proteins involved in this? Are they found to be coexisting in the fibers or in the end product, or do they promote the aggregation of AR to misfold and aggregate? Any ideas like synuclein or tau? I don't know what others.
Right. Not that we know of. So it doesn't, like the aggregates in SBMA don't stain with antibodies against tau, synuclein, TDP, FUS, so the sort of the usual players, as far as we know, are not sequestered into those aggregates. But there hasn't been, as far as I know, like sort of a proteomics analysis as to define all of the components of these intranuclear inclusions. So yeah, I can't be definitive and say it doesn't happen, but we don't know of other sort of classic protein aggregation disorder proteins that are co-sequestered with the polyglutamine AR.
Thank you.
Yeah, I guess it would be interesting to look at some of those RNA binding proteins with the prion-like domains to see if those are sequestered in, in addition to TDP and FUS. But we haven't done so yet.
So you mentioned about the small molecule designed by Jason Getzwicke, I think.
Yeah.
Could you give me some information about the molecule? Is it out there or patented, or did you guys have a dedicated screen for small molecules?
So what happened was when Jason was here at Michigan, we were working with him and Bill and Yoichi, looking for or hoping to identify small molecules that stabilize the ADP-bound conformation. Jason found a report in the literature of a compound from Fuji Chemical that had been shown to bind to HSP70, MKT077. And so that's really the scaffold, the structural scaffold for many of the HSP70 targeted small molecules that Jason has designed. So YM1, which was the original compound that Jason's lab synthesized, is really a structural derivative of MKT. He went on to show that it binds to HSP70 in sort of in a cell-free system by biotinylating YM1, and could show binding in low nanomolar range or low micromolar range. And Eric Zudervig did NMR structural analysis to define the site on 70 that YM1 interacts with. And then he subsequently made several hundred derivatives of YM1, looking for molecules that could be that have a better tolerability and pharmacokinetic properties for in vivo use. So the one that we gave to our mice is one of many that Jason has made.
I see. Okay, thank you.
Yeah.
Can I follow up with one question that just shows my complete ignorance about cell biology? Is there a reason to expect why these aggregates should appear in the nucleus? I would have expected if it's protein degradation, they would end up somewhere in the cytosol.
Yeah, yeah, no, really intriguing question. So, so the simple, or one answer would be, well, when the AR binds to ligand, it normally translocates to the nucleus. So it's actually aggregating in the compartment that one would expect it to in the presence of ligand. But the fact is that other polyglutamine proteins that are normally cytosolic also end up as forming intranuclear inclusions. And exactly why the nucleus is a favored site for that aggregation remains a mystery. Even though, and I guess the last thing to point out is that there are also cytoplasmic aggregates in some of the polyglutamine disorders, well described in Huntington, and also occurring in some of the SCAs. We don't see them in our SBMA models. So where it's strictly nuclear.
Andy, as far as I know, hi Martin.
Hi, how are you?
So actually the nucleus has a very high concentration of proteasomes.
For sure.
Yeah, so that's true. So let me follow up my questions, try to elaborate what I heard. I joined a little late, so sorry about that. So you showed that in the disease condition, autophagosomes are higher. So that means, the increased autophagosome tried to handle it, but could not. So on this branch, I'm assuming that starvation exercise will not be able to fix the problem, because that's one branch. The other branch is a proteasome degradation. So your HSP70 or HSP90 interaction with the proteins have similar affinity with the polyQ with non-polyQ. So, and then on the gel, you show that by activating HSP70, you decrease both the high molecular weight ubiquitinated proteins and the monomers. So that means you need to disaggregate so that the proteins can be degraded. So that means to me that, the proteasome is more important in this case. Is that your interpretation or?
Yeah, absolutely. I think that the proteasome is really the key driver of polyQ-AR clearance in vivo and in cells. To your point about alternative ways of manipulating autophagy, we actually did an exercise trial in the SBMA mice, in part spurred by the fact that many patients were asking whether or not they should exercise or not. And really sort of consistent with the idea that activating autophagy may not be beneficial in this model system or in this disease, exercise actually promoted atrophy.
Okay. So that makes sense, right? Yeah.
Yeah. Well, right. It does. You hate to say to a patient, don't exercise. Yeah. So I would never say that, but that's what happened in the mouse. In terms of the disaggregation, I think you're right. It's a really important and interesting area that I think is ripe for further study. We know that ubiquitinin-2, which has been proposed as a component of 70-dependent disaggregase machinery. We know that ubiquitinin-2 co-localizes with the aggregates in both cell culture and in SBMA mice. So I think that, the model that I showed where the chaperones are acting on sort of the soluble monomer may be true for some of the species, but I think it's also possible that it's acting on the aggregated species, and it's probably a mixture.
Okay. So I want to follow up that further. You showed that the Huntington, for example, affect more neurons than the androgen receptor. Is that big? What's the reason for that?
say that again.
So you said, you know, in the Huntington protein, the polyQ Huntington affect more neurons than, so the reason I'm asking,
Yeah.
than the androgen receptor. The reason I'm asking is because, you know, I'm working on HTT secretion. So unconventional secretion, we have very strong evidence to show it's indeed secreted in autophagy dependent manner. It also requires a Gaudi protein called GRAS55. So I wonder if this different kind of polyQ proteins are, you know, secrete differently so that they can affect the neighboring cells differently.
Yeah, that's a really interesting question, and certainly Huntington probably spends a lot more of the protein life in the cytosol than the androgen receptor does in a male, where there's sufficient hormone around to drive it into the nucleus. So I think that the consequences of autophagic activation or secretion mediated through some of those components, you know, it would probably be more dominant for a protein that spends more time in the cytosol, as you know. But I think the drivers of selective vulnerability are why at end stage Huntington is more diffuse than the toxicity due to the polyglutamine androgen receptor. You know, it's an unknown, and we need some good hypotheses tested. And it sounds like, you know, it would be a really interesting idea to pursue, that one that you just proposed.
Okay, well, if you're interested in collaboration.
Yeah.
Yeah.
Cool.
Yeah. All right, I have another meeting. I need to.
Okay. All right. Yeah. Thanks for the great question.
Nice talk. Yeah.
Yeah.
See you, Martin.
I see you. Nice to hear from you again. And a wonderful talk, really.
Thanks.
Just
Yeah.
fascinating.
Dr. Lieberman, I have one more question.
Yeah.
Yeah. So you talk about chaperone HSP70s. So has your lab or other people in the field looked at the specificity about, you know, different chaperone for different amyloid proteins? Like in your case, HSP70 for your estrogen receptor, and like for other protein like a tau or synuclein, whether there are different chaperone protein like HSP70 or 90 or 40, they are more like dominant for, you know, one of those amyloid protein. And how, I think you mentioned in your slides, do you just normally use a pull down assay to identify them because they bind to each other?
So let me answer the first part of the question, then I'll have to ask you to repeat the second part. Okay. So we don't know, or I don't know if if chaperone, I would assume that there's going to be some differences between clients, you know, or misfolded proteins. Again, from our perspective on the HSP90 chaperone machinery, you know, if the misfolded protein is normally associated with the 90 chaperone machinery, we might predict 70 and 90. There are a whole slew of 40s. Nobody, I think, has a great handle on why there are so many 40s. So there are going to be co-chaperones that are probably different depending on the misfolded protein and the cell type. And then if you move out of the 90 client proteins, you know, I think it's really reasonable to envision that different chaperones would play a role. You know, small heat shock proteins, etc. And then could you repeat that other part of your question?
Yeah, so the other question is, how do you identify which one is the major chaperone for your amyloid protein? Do you just using like pull down? Because you have like a polyglutamine rich estrogen receptor, And I think you know, in one of your slides, you may show some data showing the like binds with HSP70.
Yeah.
You just using pull down assay to identify them.
Right. Right. That was simply a pull down and then asking, are the chaperone machinery components there? So we just probed for HSP 90 and 70. We stripped them off, and then we rebuilt the chaperone machinery, either using a whole reticulocyte lysate, or we've also done it using purified components of the chaperone machinery. So purified 90, 70, 40, P23, and similarly reconstruct the chaperone machinery on the polyglutamine androgen receptor and get ligand binding. So that was our specific question. We didn't pull down the polyglutamine androgen receptor and do a proteomics analysis to see all the proteins that bind.
Yeah, that was a very nice experiment. Your reconstitute was, you know, reconstitution experiment.
Right. Yeah. Thanks.
Thank you.
Okay. Thanks.
Magda, I think we can wind up.
Yeah, I think we can wrap it up. Yeah, thank you.
So. Yeah,
thank you, Andy.
Yeah, that was really fun. Thanks for the opportunity to participate. This is a great, it was really great. It was really fun.
Great talk, great topic, great talk, great research. Learned a lot.
Great. Okay. All right, good. Well, I look forward to listening to future talks.
Thanks, Andy, very much.
Okay.
Thank you, Andy.
All right.
Thanks. Yeah.